I won't be very sophisticated, at least from my point of view, but I'm coming back to these issues over and over again because I see them appearing. So it seems that they are always relevant. So let me step back quickly. Well, mostly in the context, I work mostly in oncology. The concept of personalized medicine or precision medicine is very hot nowadays, and it's very much linked to this idea of finding biomarkers, prognostic biomarkers, which allow you to distinguish patients with respect to their prognosis, and predictive biomarkers, which essentially are related to the response to different treatments. And the whole idea about precision medicine is related to finding basically predictive biomarkers where you want to select patients who will be more likely to respond to the treatment which you are actually using. Obviously nowadays omics research provides a lot of data and a lot of candidate biomarkers. You have many omics variants and so on. You can have even almost anecdotal omics domains. So, okay, there's a plethora of data there, all right? Thousands, even millions, as in case of GWAS potential candidates where you can find biomarkers. So that's one of the issues when it comes to the analysis of omics data. So if you step back and look at some papers, in 2007 already people were claiming that discovery of biomarkers is inefficient. And well, there were some key points like promising initial results have been found to be unreliable or not reproducible. And then the larger process of discovery can be slow and inefficient. Studies are unreliable because of problems from chance and bias and so on. Okay, so it's, 11 years ago? So people already there were seeing that we are sort of like, we are having enough data to mine it, but it seems that we are not doing this very well. Then you go on in 2011, and then you see that someone again picks on this point: publication bias, hidden multiple hypothesis testing, distort the assessment of the true value of biomarkers. And then 2014, 3 years later, major issues to be resolved. And I have made, just for the sake of another presentation, another search last year. And in 2017, you also find the same story. Discovery of biomarkers is inefficient. And they mention poor methods, selective publication, incomplete reporting, spin in titles, abstracts, and so on. So you can see over the period of over a decade, same things were reported. So it seems that there's not much improvement there. And that's why I'm trying to preach the same story over and over again, although it may be boring for some of you, because the point is that, especially for young researchers who start their career, these things pop up. So what are the errors which are plaguing the discovery of biomarkers? Multiple testing. This is one of the most important things. In the previous presentation, we saw already this term p-value, 10 to minus 40, but you have to be careful because p-value is related to random processes, and the play of chance is deceptive. I'll give you an example. Focus on association measures for prognostic biomarkers instead of discriminative properties of these biomarkers. Faulty study designs, sample selection, and so on, improper validation of the biomarker studies. And then you have also publication bias, because you see only these papers where, well, yes, this locus is associated with the disease, but you don't see 5 other papers which couldn't confirm that. So that's the same story as with respect to the clinical trials, where publication bias was actually leading to the discovery that the effects of some treatments were overestimated. So let us look at a couple of these examples. So really, the play of chance is deceptive. And I will give you an example from clinical trials, well, not from omics world, but I will give you some calculations which will tell you that with omics, with biomarkers based on molecular biology experiments, you have an even bigger problem. So this is an example, very simple, 2 trials. 2 clinical trials were carried out to test a new treatment for age-related macular degeneration, so for a disease of an eye, okay? And then each trial recruited about 500 patients, one in the US and one other in Europe. So actually these 2 trials led to registration of this drug both in the US and in Europe. And then overall, there was a beneficial treatment effect. The famous p-value was less than 1%. So as I said, well, very well for regulatory agencies. Everyone was happy. Okay, they read the disease. And then one of my colleagues looked at this data and looked at the exemplary analysis of the result of the treatment by astrological sign, okay? So you just look at 12 groups you subdivide your patients according to their astrological sign. You look at the results of the proportion of the responders versus non-responders in each of these subgroups. And you see in Libra, you find, of course, the testing is done without any adjustment for multiple testing. So you see for Libra, there's a higher proportion of responders for sham, so for placebo, than for the experimental treatment, right? Okay, well, biologically and medically, this is, I don't think it's very much, expected, right? I'm Libra, so I'm doomed when I get, well, with my age, I mean, when I get age molecular degeneration, well, I'm screwed, sorry. All right, fine, you might say, okay, fine, well, we always say we should repeat, we should actually reproduce these results. So this is your European trial. Look for the US. Yes? Libra again. It must be true, right? I mean, 2 independent studies, 2 independent analyses, you see this. Well, that's how we underappreciate the play of chance. This is due solely to the fact that, of course, this analysis is incorrect, that these were the tests which were done without taking into account that we do 12 tests each and every time, and just the same significance level was used in each of the tests. Now, as I said, this is the clinical trial, but let's think about omics research, okay? What is the multiple testing issue? The fact is that if you multiply test, for instance, you split your data set in many ways, and even if in this data set there's no signal, then if you do many tests, you are sure to find something. This is just a calculation, theoretical calculation. So assume that you have m independent biomarkers. So it means you have like m, you split your data in m subsets. Assume there's no difference, for instance, between cases and controls, right? So we have 2 groups, and then you look at, for some biomarker data, and you look at cases and controls, whether there's a difference for this biomarker, and then you take another biomarker, you look whether there's a difference between cases and controls, and for another. So for instance, if you take one biomarker, and if you test for the difference between cases and controls at 5% significance, of course, the probability that you will falsely find a significant result for this comparison is just 5% if you do just one test. But if you look at 2 biomarkers, even if for neither of them there's any signal, any difference between cases and controls, well, the probability of finding one of these tests for one of the biomarkers finding the significant results is about 10%. If you take 10 biomarkers, it's 40%. If you take 100 biomarkers, you are guaranteed to find something, okay? Now, 100 biomarkers is peanuts. I mean, if you look at the GWAS studies, there are millions of locations which you are testing, right? So you should take this into account. But someone might say, okay, fine, cool. Let me, well, forget about this adjustment. Independent study. I will do this, and even if I make a mistake here, I will do an independent study, and then I will be done, right? Well, not quite. This is the probability of doing 2 studies, 2 independent studies, and just looking at the number of biomarkers and the probability that you will find a biomarker falsely in one study, and then you will repeat it in another study. This is like a case of these 2 trials. So surely enough, if you look at enough biomarkers, look, if you have 1,000 biomarkers, and that's not much by omic standards, you have 70% chance that you will do one study and then another one, and in both studies you will find at least one of these biomarkers consistently being picked up, okay? Of course, it's 1,000. If you calculate it for 10,000, like genes, well, you are guaranteed to find at least one biomarker which is independently found. If you are not careful about statistical analysis, that is, if you don't correct for multiplicity. Now, anyone might say, oh, yes, we know that, right? Well, yeah, take any paper in biological or medical journal and see how often you will find the phrase, we have corrected for multiple testing in our analysis, right? Even if they did, well, you may not actually see it written, okay? So you never know, all right? So that's one of the issues. So you should bear this in mind and always, especially in omics analysis, correct for multiple testing. Now, this is a very harsh so-called Bonferroni correction. You take your significance level, which is typically 5%, and you slash it by the number of tests you do, okay? So if you do your, let's say, 100,000 biomarker study, you should take 5% divided by 100,000 as a significance level for each individual comparison, okay? Fine. one of the things which I see over and over again, despite the fact that over years the same problem is reported in these papers over years. Now, another thing which another problem is that usually what is done is just association test. So imagine you have two populations, cases and controls, and this is like a biomarker level, all right? Now, if you test in these two populations, if you just do if you collect two samples, you test the mean, and then you will do comparison, you will find statistically significant result here. Why? Because means are different. There is an association, right? If you do it here, surely, well, your p-value will be even smaller. And also here, even better. But only here, it is meaningful to think about discriminatory properties, right? Because the overlap between these two populations is small. So if you put a threshold on the biomarker here, then you will have a nice probability of distinguishing between the controls and cases, right? But if you put any threshold here, there will be a large proportion of cases who will be indistinguishable from controls using their values of biomarkers, because the overlap between these distributions is too large, right? So the fact that you do a t-test or a test based on whatever the model, and you say, oh yes, there is statistically significant difference in the means between the two populations, it doesn't mean that this is a good biomarker, for instance, for diagnostic properties or prognostic properties. And you see it over and over again. For instance, this is a classical gene signature in breast cancer based on 70-something, 72, I think, genes, a signature for looking at the metastasis, probability of metastasis in breast cancer patients. So these are results in New England Journal of Medicine. This is the key table. These are the patients who had distant metastasis within 5 years, and those who were free of metastasis. And here you have two groups: poor prognosis and good prognosis, based on this score, which was created from 72 these genes. All right. So if you look at this table, and if you look at the measure of association, which is the famous or infamous odds ratio here, it's huge. It's 15. You rarely see this in clinical medicine. So obviously, you say there are 15 times higher odds of metastasis for poor prognosis signature than for individuals with good prognosis signature. 15 times higher odds, right? So it means that, you know, you have poor prognosis signature, obviously you are more likely to have metastasis, right? But is it good for discriminating? So if you now do the test at the baseline for the before treatment, or just at the one point for the individual with the breast cancer, and if you want to predict whether there will be metastasis or not, is this a good marker? Well, here you look at the sensitivity specificity. Sensitivity is 90%, but specificity is only 60%. So I mean that this test is inspecific. It's in 40% of the cases, it's not specific, right? So it means that you actually, what does it mean? Sensitivity is fine. It means what is the probability that if the individual has a disease, or whether the individual is prone to have metastasis, what is the probability that the test will give you correct result? Will say, okay, this patient probably will have the metastasis. Well, okay, it's 90%, fine. But if you say, okay, this is individual who does not have a high risk of metastasis, with which probability will have a negative result for this biomarker, poor prognosis, good prognosis, it's only 60%. So for 40% of the women, you will actually get a wrong result. You will say, oh, you may actually you may be prone to the risk of metastasis, but this means that you will treat these women perhaps with the toxic chemotherapy. And this is not surprise, because you can show that in order for a binary biomarker to have a good discriminatory properties, your odds ratio doesn't have to be 15, it has to be in the vicinity of 100. How many of you saw 100 as odds ratio in clinical medicine? I haven't. So that means that when you look for discriminatory properties, when you look for a biomarker which is served as a diagnostic marker or as a prognostic marker, you shouldn't look only on this association, but you should really look at whether you can discriminate one population from another. But rarely, you see all these papers about biomarkers, they say, oh yes, we had this p-value of 10 to minus 4. There's a difference between the mean level of expression or whatever. Well, that's not the end of the story. If you go to the clinic, you want to have discriminative properties correctly, okay? So the hint, do not overinterpret association measures. If you really are after something which you want to use for diagnostic purposes, look at discrimination-related measures. You look at sensitivity, specificity, you can have these ROC curves, you can look at the area under ROC curves, whatever. But you should look at that in addition to the association tests, all right? Whereas mostly what I see is that people stop after association tests, okay? Another story. Omic technologies are really sophisticated and impressive, right? They use advanced scientific principles, complex instrumentation. They produce massive amounts of data, so-called high throughput and so on. So these are wonderful machines, probably not even understood by their developers, right? So what it means is that these things are vulnerable because they are highly sensitive. They have systematic, you can see systematic effects due to the time, place, reagents, personnel. Everything can be visible. I could give you anecdotes about effects I have seen over my career where people were seeing difference and they were excited about difference, and it appeared that the difference was simply due to the fact that they were putting the samples on their laboratory well too long, plate too long, okay, and then activity of what they were studying was actually dying off, so they were actually having these problems. Reproducibility therefore can be easily compromised. You do the experiment in one lab, in another lab, and you don't see the same results. Variability can be considerable, and it can be due to very different factors, and it's difficult to control these factors. And naive data analysis can lead to erroneous conclusions because of that. You have to be careful about how you design these experiments and how you analyze them. So this is an example. It's actually liquid chromatography mass spectrometry data based on metabolites. Well, they just took the same sample. So they took extracts of drug-sensitive and drug-resistant strains of this organism, and then they just split it and they just did it twice, okay, in 2 time blocks. And here, this is like an average measurement which they obtained for these different runs on one day and on another day. Well, I see a pattern, do you? This is one day, this is another day, right? What happened? Everything could happen. I have seen these trends everywhere. In mass spectrometry, the technician comes on Sunday to, let's say, to maintain the equipment. Well, all the settings of the equipment goes back to what they were usually before. And you see, next week you have, on average, higher mass spectrometry reads than it was in the previous week. Why? Well, there was maintenance, and maybe the detector was somehow cleaned, and therefore it gets better readings. So yes. Well, if you look at the data like this, and then if you cluster your samples, you see this. This is the cluster of one sample, that's another of samples. Do you see any, well, what happens is these are indicators of the samples. Obviously, you cannot see them, but you clearly see a pattern, right? This is one day, this is another day. This is the structure of your data. If you are not careful, well, this is what you will discover. What you need to do is you need to get rid of this time effect. Now, there are simple methods for there. They are more advanced as well. And if you do this properly, now you see the story, right? Now, if you start to do the cluster, and then you will look at how samples are clustered, you see these are the sensitive samples, and these are the resistant samples. So if you remove the time effect, then you will see that you can distinguish, actually, based on these values for different metabolites, you can distinguish these different samples from each other, right? But if you do just naive analysis, take the measurements as they are, and you will compare, what you will see a difference between samples on day 1 and samples on day 2. So, well, first of all, you have to know how to design experiments. So statistical design of experiments tool can help you. You should always randomize the order of your samples. I know that you know this, but I would like to remind you, always randomize. Don't do this in chronological order because you will be confounded by the time effects. And then there are methods which help in removing confounding, which may be you might have collected the data. There may be already confounding, but statistical analysis can help you with. But you have to be aware of this, okay? And finally, something very obvious, but you also know it. You should avoid overfitting when you develop, for instance, a signature, all right, a gene expression signature. This is, let's say, this is training data set, and then you have testing data set. This is complexity of your parameters. So let's say you did a signature from 1 gene, 2 genes, 3 genes, and so on. You make signature more and more complex. What happens is that you will see, this is the predictive accuracy. So your accuracy of your prediction, if you increase the number of, for instance, genes included in your signature, it will go, and surely enough, it will hit 1. You will be perfectly able to predict, to discriminate between cancers and controls in your training data. But if you look at this, if you take take your signature and you apply it to the testing data, you will see this, that if you start to be overly complex in your signature, you will start to lose your prediction. Why? Because the more complex your predictor, the more you start to look at the random error which sits in your training data. And then if you start to apply this procedure to the independent data set, there will be a different error structure. And then you will associally make errors, right? So you should have really a proper validation procedure. And I will not go into details, but you see also that, for instance, these guys from this signature, when they did the validation study, they actually included samples from the original training data into the testing data. So in fact, they validated their signature partially on the same data set on which they developed the signature. Okay. Now you would expect that New England Journal of Medicine should know better when accepting papers for publication, but this is a top medical journal, and they also allow these two things to pass. So don't believe in the referring system that it will actually be able to spot all these errors. So this should be a rigorous development process. You identify signature, you have internal validation, so perhaps using cross-validation based on your training data, do external validation, proper external validation, take independent data set in the same population, freeze the signature, and so on, and only then try to talk about clinical utility. We all know this, right? Yes? I mean, I could see you smiling, right? Why, therefore, we see these papers every 3 years published? Well, because you know, but probably some others don't. That's why I tend to talk about this. So this is the usual quote which I end up the presentation with. So I'm available for discussions later on. Thank you. There are usually none, so don't worry if you don't have any. And please talk to us before you die or your experimenter dies, okay? Thank you. Well, look, first of all, there's no data, no experiment which does not need statistical help. And then, well, unfortunately, my presentation is gone. On the presentation, there is, for instance, email of myself. So if you don't have anything there, well, mail me, all right? And then you can actually get some indications from me. And I can, for instance, point you to someone who is in your vicinity, all right? But mind you, there's no situation when you have inference based on data where you would not need statistics, because when you do inference based on data, it means that you are vulnerable to random error, and statistics controls random error. That's the bottom line.
Yes, so just recently there was in the last issue of Nature, there was an article suggesting that in the areas of the humanities, the scientists should also look for ways of introducing a deep replication of the analysis. One of my colleagues commented on Facebook that perhaps in theology that would be an interesting experience.
Well, yeah, I could imagine some research done with statistics and theology as well. Here's my email. Thank you very much.
Thank you. Does this work? Okay, it's fine. Good morning, ladies and gentlemen. My name is Konrad Palgac, and as already said, I am a PhD student in the Department of Biostatistics and Translational Medicine here at Medical University of Lodz.
And I have a pleasure to tell you about our novel approach to normalization of qPCR microRNA quantification data. So first of all, what are microRNAs? MicroRNAs are short, non-coding RNA molecules, usually counting between 20 and 24 bases. Their main role in cells is to inhibit gene expression, mainly at transcriptional and post-transcriptional levels. They are ubiquitous. They are found virtually in all human tissue samples and are also circulating in human serum and thus can be quantified. qPCR is a widespread method. I think almost all laboratories perform them. This widespread use could be attributed to its simplicity, reproducibility, its cheap price, and that it's pretty fast to run, for example, compared to RNA sequencing. And the intrinsic nature of qPCR is that it's a relative method of measuring the abundance of genetic material. So it needs some kind of a reference gene. So we could compare different gene expressions to the reference gene, and then this difference will be compared between samples in a method called delta-delta-CT. Many recommendations have been already proposed to for qPCR reference genes. I listed just 3 of them, so not many. Some of them are tissue-specific, some are not. But no such recommendations for microRNA reference, whether the experimental validation, at least in qPCR, from serum samples. So this leaves a room for us to maybe propose a new method of finding such reference gene, because the problem might not be that the microRNAs are not stable in serum, because they are, but in the way of how we propose candidates for them. So our goal is to identify a single microRNA or a group of microRNAs that we can use as a good microRNA references. To that end, we gathered 11 data sets from gene expression only database. We accessed it with the query on the right on October 1st, 2017, I think. We excluded data sets that contained less than 170 microRNAs, less than 5 samples, and more than 20% of missing data. This is to ensure that our analysis is robust and algorithm work properly. We used 3 different normalization algorithms: BestKeeper, Genome, and NormFinder. They all work in a pretty similar way. They process a single data set each time and assign to each microRNA a single value, which is their normalization score from 0 to 1. The lower the score, the more suitable the microRNA to be a reference microRNA. Since the distributions of these normalization scores differ between algorithms, we made the algorithm output actually a ranking of microRNAs from best normalizer to the worst reference microRNA, and not the raw normalization scores. This is to ensure that we can aggregate the results and compare them between algorithms. So first of all, we wanted to analyze all single microRNAs, and then we wanted to analyze combinations, all possible combinations of 2 and 3 microRNAs. And we did it by treating all combinations, each combination, as a single microRNA. So we assigned a single expression value to each combination. And this single expression value was the geometric mean of the components of the combination. So in the process, we could add those combinations one by one to data set with single microRNAs, run the analysis, report the normalization rankings and scores, and remove the so-called combination and add another one. So in the data set, this is to ensure that only one combination is at all times in the data set, so the combinations do not interfere with each other. These are results from our single microRNA analysis. So the intensity of this on this heat map is what represents the mean normalization rankings from 3 algorithms in a single data set. Data sets are in columns, and in the rows are microRNAs. They are ranked from top to bottom, from best reference microRNA to worst microRNA reference. As it turns out, on the average, MIR222 was the best reference microRNA, soon followed by MIR16, MIR19B, and MIR484. So we concluded that that our results from single microRNA analysis are heavily weighted by the popularity of microRNAs in microarrays. So the more common the microRNA is on the microarray, the more probable it is that it's going to be a good reference, just from the sheer fact of that it occurs a lot. And also that no single microRNA would be a good reference microRNA, because the gray color are missing data, and single microRNAs tend to be missing sometimes in some of the experiments. So a single microRNA wouldn't be a good reference, because we need the reference to be present in all experiments, so we can compare the results. Okay. So the idea of creating combinations stems from actually this heat map. So this heat map presents raw expression values of microRNAs presented on the previous slides, and as you can see, they differ inside the data sets. So this is very much different from this. And our idea, we hypothesized that maybe if we take the average of 2 or 3 mirs from this raw expression value, we actually could create a better reference than the single microRNAs. And as it turns out, we were right. This table represents average normalization rankings in the data sets. Each dot is a single data set. And as you can see, pairs, triples, and also just mean expression of all microRNAs are a better reference than single microRNAs. There is no difference between pairs and triples in that regard. So the next question we ask ourselves, okay, they are on average better. So the combinations are on average better, but are top combinations better than the best single microRNAs? And that's actually true. This heat map shows ranked best microRNA references from the combinations of 2 microRNAs and single microRNAs. So red color is combination of 2 microRNAs, and blue color is just single microRNAs. As you can see, top is the best reference and the bottom are the worst references. As you can see, the red color is on top of the blue color, so it means that in each data set, yes, combinations were actually better than single microRNAs. So then we ask ourselves the question, so how to efficiently create those combinations? Well, we thought that maybe we can just take a fraction, maybe top 10 single microRNAs from each data set, and create combinations from that top fraction of single microRNAs, and we could create a good pair or a good triple. So we ran that analysis. We wanted to know how big of a fraction of a single microRNAs from the original file we have to take to create those top 10 pairs and triples. And as it turns out, often we have to take almost the whole data set, as in data set number 2 and 3. So this is not an efficient method because of the calculation times that take to calculate those. So our next thought was maybe we can just introduce a universal combination of 2 or 3 mirs. So we actually gathered occurrences of single microRNAs in top 10 combinations of 3 and 2 mirs in all data sets. So we wanted to know whether we can create a combination of 2 and 3 that would cover all of the data sets. As it turns out, it was not feasible because of the sheer number of hits, in those top 10, 75 and 96 in combination of 3, and it was not possible to find a combination of 2 and 3 that would cover all the data sets. And this is also strengthened by the idea that if we just take a random combination of 2 and 3, we ask ourselves a question, so what is the chance that if we take just a random combination of 2 and 3, that the combination is better than each of its components? And it appears that, yeah, it's pretty good, all in all. We have high chances of that, but we cannot ensure that the combination is always better than each of the components. So we figured out that we have to take another approach, and our idea now is to propose a group of multiple microRNAs, for example, given 5, from which we can derive a good reference in the form of a combination of 3 mirs or a pair of mirs. And additional mirs on top of the 3 that we calculated right now would cover for the possible missing data in our experiments, in different experiments. So there will be So we have to find a group of multiple mirs that we know that combination of them, combination of 3 out of 5 of them will be a good reference. Okay. So sum up. The combinations of 2 and 3 microRNAs are in fact better references than a single microRNA, and combinations of multiple microRNAs which cover for missing data are needed to provide a robust and reliable reference microRNA for qPCR data from serum samples. Thank you very much. Sure. I want to. Okay. There's a problem with our microRNAs because there are many missing data. Can you update? I wanted to show you this. So this is a gray color is missing data.
I don't understand that, but well, that's my point. I mean, if you
Yeah.
say, okay, so you mean that you want to have like 5, so it means that you cannot make an average, right, of 5.
No.
One component of this 5.
The idea is a little bit different. So we want to have a combination of 5, and if you have, for example, and if all of 5 of microRNAs are present, you take combinations of 3 and 5. You can run the algorithm, which is pretty fast because the number of microRNAs is small, and you can rank them, these combinations of 3 out of 5, by the algorithm, and just take the best one, and you are ensured that yes, the best one is the best combination. So we take the combination of 3, so the average of 3, not of 5.
So you mean that, for instance, but I understand that not all of them are present, right? So you mean that you want to again just look at the 5 which are always in all data sets?
No.
So what happens if out of these 5 you have just 3?
So this is our approach we want to do, and we want to find such 5 that are always, that at least 3 of them are always present in all the experiments.
I get you. So the problem I have with that is that,
Yes.
well, I suspect that you may have missing microRNAs, right? Missing because the level of detection was relatively low level of detection which was small. I suspect that the missingness is really informative missingness in the sense that it's related to the fact that the expression was so to me, the point of trying to cover up this, I mean, the idea that we want to, find a group of 5 which are always, or 3 of which will always express, and then you say, okay, in this way I will cover the 5 that I will try to, let's say, compensate for the fact that, in our case we may have missing data. I think it is okay if you assume that the data for some of these microRNAs are missing just at random. Sometimes it happens they miss it, but I don't think they are missing at random. I think that at some point, on the fact that they are missing it is because in the data set, the level of expression, for instance, was very low, was not detected or something. And in that case, well, the fact that anything what is observed probably overestimates, or it has completely different mean value than what you would expect from the missing values. So I think to sum up, I mean, I can see where you're trying to go, but
Okay.
this is okay if you assume that this fact that you have this gray color is not related to the level of expression of your microRNA.
Generally on the microarray, right?
Yeah, but if it is, well then whatever you observe and whatever you average, it cannot actually compensate, I mean it cannot represent what has not been observed. And in this way, respect, I don't think how we can treat it. This is what
Well,
I
if
think.
so, that's why we have a lot of data sets, and we try to ensure that our microRNA reference is always at least 3 out of 5 are present, right? If the microarray is generally low in quality, yes, so the reads are pretty low, right? But that would be a question of whether it is a good whether it is a good experiment to base your analysis upon, also, right?
Okay, but that's a quality score rather than just about the reference value, right? So, okay, anyway.
I mean, that's a valid point. Yes, what happens if all of your reference microRNAs are not present in your data set? and yeah, that's the valid point. So we try to cover up for it by gathering as many data sets as possible and ensuring that, well, we try to find the most stable microRNAs still present in all of them. And we try to ensure that, well, in all the previous experiments, at least published, there they occur, right? And they are not below the level of well, qPCR detection rates, right? Okay. That's a very valid question, yes. So your concern is, I guess, sorry? Universal. That's so your concern is that the biological conditions of, I guess, people that would be involved in study would affect the reference, right? So this is why we try to find, well, if you know that some kind of microRNA is adjusted in your disease, right, and it's changed, then you can exclude it from reference, for example, yes, and maybe, yeah, that I think is the idea. But this is a valid concern, and that's why we try to gather more than one microRNA. It's very rare that I think a group of microRNAs is just lowered or, well, has lower or upper expression, and this type of microRNAs, since we have studies from different diseases, these 11 studies actually involve studies with people with breast cancer, prostate cancer, some neurological diseases. So the reference that we will find will probably be independent of the disease. So they will be disease independent. That's at least what we hope for.
If we assume that the microRNAs are biologically dependent on each other, that they are not necessarily.
Yeah, they are not necessarily, yes.
But it's a long discussion, and then you can take offline. So let's thank our speaker again. Today's session is Dr. Liana Bukowski from the Nicolaus Copernicus University in Paris. He's a physicist by training, so we have an interesting here now.
Yeah, we can. Thank you very much for your introduction. We can switch the gears a little bit so I can see my presentation just in a minute. So, okay. So, as you said, I'm a physicist who works mainly with ophthalmologists, and today I'm going to talk about the three-dimensional assessment of crystal lens opacities in cataract patients using a novel device which is called long depth range swept source optical coherence tomography. And from the physical optical point of view, we can see that the eye is the optical system, which is composed of few elements, and they have different optical properties. And the quality of vision, the image sharpness that's projected into the retina in the fovea is limited by two factors. And the first factor is actually, so in the ideal case, we have sharp image. You can see, so when the light is focused exactly at the fovea. And the first factor is aberration. So this is geometrical disruption of the intraocular surfaces of the interfaces, and this can be corrected by the glasses or by the optics. And also, and you can see that this causes the image to be blurred. But also there's another factor which is called intraocular scattering, and this causes that the image is the contrast of the image is much lower. And the most important is that the scattering is affected by the opacifications, and the opacifications mainly are related to the crystalline lens, and this disease is called cataract. And we can see that we can have different forms of cataract. Then they can be formed in the nucleus, they can be formed in the cortex, or they can be formed in the subcapsular regions. And so the doctors can somehow differentiate between different features. And there are different methods that can be used to assess and to quantify the opacifications or the cataract. And in the clinical, usually in the clinic, you can use visual acuity test, ophthalmoscope, or slit lamp. But also there are like imaging methods. However, some of those methods, they can use only, they don't use like comprehensive information. They can use only two-dimensional information, or they can produce like general parameters that tell us something about the opacifications. And what we are going to do, we are going to present you the three-dimensional visualization of the crystal lens opacities in vivo. We want to identify the features using the method which is called OCT, and to develop some quantitative data analysis strategies for opacity grading. And what's OCT? OCT, optical coherence tomography, is the method that enables us to detect the light that is reflected or backscattered from the intraocular structures. And this is the principle of operation is similar to ultrasound. However, use light except for the ultrasound, and we can extract the depth information about which tells us how much light is scattered from those structures. By scanning the beam over in transverse direction X and Y, we can either obtain the cross-sectional image or three-dimensional image. So we can have volumetric data with very high resolution, like micrometer resolution. Of course, we have actually the OCT is developing so rapidly that if you have very high-speed system, you can even image in time, and this is called 4D imaging. So usually OCT is used in the diagnosis of retinal diseases. In the US, actually for the anterior segment, OCT is actually regarded as the investigational device. So it's not still like accepted by the by the clinicians. And so there are several challenges if you wanted to measure the anterior segment of the eye. There are different challenges. Anterior segment is quite deep structure, so you need much longer depth range in your imaging. And the other is you need to have ability to perform quantitative analysis, and you need to have very high contrast images to visualize differences in scattering properties, and you need to have very high speed system in order to perform in vivo imaging. And we addressed all those issues, and we developed the system that was deployed to the University of Murcia, to the Laboratory of Optics of Professor Artal. And this is how this system looks like. You have the interface, the screen, and then there is detection and acquisition software. So the system operated at 50,000 depth scans per second, and I can skip this because of the short time. So we optimize actually the system, and you can see that we use also different objective lens in our interface, and you can see that the longer focal length of the objective lens gives us much better image quality here, and this is more or less resistant to the eye position differences. This is important from the clinical point of view. And we also optimize the drop of the signal. The drop of the signal tells us how quick signals we can visualize with that, and you can see that there's practically no difference. It's practically stable, like constant here over the 12 millimeters, and this is actually what we wanted to get. And so we image 35 eyes of 21 subjects with or without opacifications. Some of them were diagnosed with cataract, some of them not. And they actually, right now we have probably 50, something like that. And we were able to obtain high definition cross-sectional images, as well as three-dimensional volumetric data sets of the anterior segment. We used no dilation for that. And what can we do with these three-dimensional data sets? So we can have like en face image. This is like photography. We can segment the pupil only, because the light doesn't penetrate through the iris, and then we can play with it, right? We can segment the interface. We can segment the nucleus. We can segment the anterior interface, posterior interface of the lens, and then we can visualize different parameters. We can generate different projections. This can be maximum intensity projection, maximum intensity depth projection, some voxel projection, or average voxel projection, and you can see here the projections of the healthy eye. You can see this posterior capsule projection of the maximum intensity, the nucleus, and also the cortex. You can see here some kind of like star-like structures, and these are called sutures of the lens. And we can show you how those images are demonstrated in different cataracts. And you can see here also how these strategies are visualized in this movie. Here you have volumetric rendering of the image, and here is the fly-through. And then after that, we can do. After that, you can see in a moment that we will have the segmentation of the iris. Here is the lens is somehow the pupil is segmented. The lens here is like false color coded, and then after that we segment those substructures of the lens, and then we can generate different we can color map actually different structures. Here is the anterior subcapsular, posterior subcapsular. Then you have cortex in red, and in yellow you will obtain the nucleus, and you can generate different projections from there. And so we imaged the cataract suspects. These were people who were quite old, and they were not diagnosed. But even though we were able to detect different structures, you can see here, for example, here hyperreflective region going from the periphery of the lens to the center of the lens, to the pupil. And this is clearly visualized here. And you can see that the advantage is that we have three-dimensional visualization of this. We can have also inhomogeneity in the lens, and also the other is, so this is like hyperreflective. Here we have hyperreflective structure, and also you can see that the nucleus here is hyperreflective. And this means that the scattering is higher, and this means that the opacity, actually, they already have opacities. And the other are cortical and nuclear cataracts. And you can see here in cortical cataract what are the changes compared to this healthy subject, young healthy subject. So you can see the changes of the optical properties of the cortex as well. Here you see the vacuole. So those structures are called spokes, cortical spokes. And here is the vacuole also, and here is the advanced nuclear cataract. And this is, you can see the compared to those images, you can see how big differences in the scattering in the signal in the nucleus. And this is clearly visualized. This is one of the most prominent cortical cataract patient. You can see the radial spokes that are affecting from the periphery to the center. They are also in the anterior part and posterior part. So okay, so this I will probably skip this. And what the doctors do usually, so they do slit lamp examination, and they have the chart, and this is called they do cataract grading. So they have the chart, and this is the lens opacity classification system 3, LOX 3, and they compare the image of the patient to the particular image in the chart, and they can score, they can grade the nucleus opacity, the cortical part, and also the posterior subcapsular. And we did similar stuff with our three-dimensional data, and you can see the examples of those different grades, and in cortical cataract, nucleus cataract, and the posterior subcapsular cataract. And you can see that in cortical cataract, when the cataract is more advanced, so more and more pupil area is actually affected. It's clearly visible here. Here also we have depth information. In nuclear cataract, you can see that the average signal is higher with the larger grade, and also the posterior subcapsular cataract, you can see the more and more area is actually affected. And so this can be easily quantified, and you can see that we quantified, and you see this number like integrated intensity or the intensity median, and you can see that with the grade it actually increases. So OCT can be regarded actually as some kind of, can also provide some quantitative information. At the end, I want to show also that it's not only cataract, that the crystalline lens that we can image, but the transparency of the ocular media can be also affected in the cornea, but also in the largest part of the eye, which is called the vitreous. And vitreous is usually perceived as the transparent part of the eye, but there are some diseases like vitreous hemorrhages, detachment, and floaters, which also demonstrate the opacities. So this is our last result, and you can see here that we can actually visualize, even though this is transparent, from the anterior part of the vitreous or posterior part of the vitreous. Our system is so sensitive that we are able to visualize this vitreous. And the question now is for us, actually, is to what's going on here. So this is the project that we are starting actually, and you can see here that this is the proof that the vitreous is actually very transparent. However, we will try to do that. So we are very happy about this. So to summarize, we demonstrated volumetric long depth range OCT, and that enables visualization of the microstructural changes in the crystalline lens. And this allows to enhance the contrast in the images. And also this might be useful in the high resolution evaluation and management of crystalline lens opacities in cataract patients. And this may help probably in diagnosis and grading the cataract eyes. So at the end, I'd like to thank our team and our collaborators from Spain. And so the next, we want to extend our research team into the Technical University of Catalonia and also BMR Institute. Those guys are actually experts in vitreous imaging. And and I'd like to thank also our sponsors. And I'd like to thank organizers for having opportunity to speak to you today. Thank you. So yeah, I forgot to tell it. So we use 1 micron. So the center wavelength is 1 micron. Actually, at the stage of designing the system, my collaborators actually said that they would like to have shorter wavelengths, but the lasers are not available at this. So we decided to use 1 micron. And but actually, to your first step, so I've been at the largest conference, ophthalmic conference, and this machine learning and all those stuff with the data processing. This is very hot topic now, and they try to diagnose without any need of the doctor. But I'm not sure if this is the future. so there are these tendencies actually in ophthalmology and visual science.
It's quite impressive from a technological point of view, but my question is, okay, so in a clinical context, right, where do you see it? Because I don't see the placement of this, because in a sense, what you have shown is that you showed the images of average images which were, let's say, overlaid with the grading system which they use currently. So my question is, will this be like a tool based on which they will still use their old grading systems and so on? And if so, then the question is, have you looked already at how well you can actually discriminate between patients with respect to grading based on their image?
Yeah, this is still to be done. So actually, the idea is to have some kind of methodology to have because adjusting the images to this chart is actually subjective. But what the doctors would like is to have a more objective tool, right? And so this is quite good because you have 3-dimensional structures as well, and you can have quantitative information in one tool, right? So still, I talked to some doctors. So some people told me, I'm not an expert in ophthalmology actually, but some people told me that actually this LOX3 is awful. So but it's still in use, right? It's still in use. It's quite old, but it's still in use. So, and the other thing is that the idea behind this was actually, if we have the 3-dimensional information of opacity, maybe we can generate some kind of goggles. So this was the idea of this CCAT project of Professor Attal. Maybe you can generate some kind of goggles, and they will modulate the light intensity that is projected. So you can pass this opacity somehow, and you can have the full image on the retina, right? So, so this is the idea.
Okay, we have to stop now.
Thank you.